So our last two factors that can play a role in microevolution and thus change gene frequencies are sexual selection and natural selection. Natural selection is something we've already kind of talked about at length. Just here's some more notes on it to reinforce what's happening. Obviously, if you have some environmental condition that you have an adaptation that's not favorable for, then you're not going to be passing on genes. If you do have an adaptation that benefits you in an environment, that gene that gives you that adaptation gets passed on with a greater frequency. The other factor that can affect gene frequencies is a sexual selection. So it's a kind of natural selection. It's along the same kinds of lines, but it doesn't always mean that a partner gets selected because they really are the most physically fit for a given environment. If you think about something like male peacocks, they're beautiful and they have the huge tail and the bright colors, but that really puts them at a disadvantage from a survival standpoint. They're much more likely to be eaten by predators, be unable to escape from predators, be unable to hide from predators. But females find the bright, showy colors and the large tail to be attractive. So even though you might get eaten at a younger age, at least you have offspring. And that's what's really important. Remember, in class, we were trying to really hammer home that point that it's survival of the reproductively fittest. And something can put you at a huge disadvantage as long as the opposite sex finds it attractive and is more willing to mate with you. So sexual selection is based on phenotypes the opposite sex finds attractive. Therefore, certain goals for attractiveness get passed on to future generations, whether it really helps you to survive in the environment or not. It is all about reproduction, so be aware of that factor as well. So a couple of things to think about. Remember when we started in on these slides, I said that we had an equation, the Hardy-Weinberg equilibrium, that if we could satisfy that equilibrium and get to 1, it would go a long way towards disproving evolution. But some of the things that the Hardy-Weinberg equilibrium say is that you can't have struggle. There can't be mutations. The only way we can satisfy this equilibrium and get to 1 is if there isn't any migration, there isn't any mutation, if there are no catastrophic events, if the population is infinitely large, and if mating is completely random. So here are some review questions, some things to think about, and these come, I believe, from the end of the chapter. So if you're having trouble seeing these slides online, just go to your chapter review at the end of chapter 13, and you'll see these questions. And so the questions here regard that Hardy-Weinberg equilibrium and how these real-life situations disrupt it so that we can't get to 1 with that equilibrium. So think about what factor, non-random mating, infinitely large population, no mutation, no migration, things like that, those things back on that slide, how these 3 examples violate those situations. So think about couples who have a disease but decide not to have children. How does that violate Hardy-Weinberg equilibrium? Or another way to think of that is how does that fit into the 5 scenarios we mentioned previously? So if couples decide not to have children, then the mating is apparently not random. They are making a decision not to mate, and that takes the randomness out of it. What about members of a close-knit religious community that are forbidden to marry outside of that community? What's happening there? Well, if we think about the bottleneck effect and the founder effect, obviously this isn't an infinitely large population. Look back at the little picture we had of the penguins. If there is a gene in that population, that small population is bad, then it's going to get larger in proportion. If there's a gene that's good, if that person carrying that gene gets killed in a farming accident, well, that's just gone, and you're not going to have new genes come into that gene pool. So the biggest problem with that scenario, as far as Hardy-Weinberg is concerned, is the lack of an infinitely large population. And if we look at an emerging infectious disease that kills individuals with a certain blood type, you know, at this time in our history, we don't have any diseases that just kill blood type A or just kill blood type B. But what if all of a sudden something popped up that killed people who were of a certain blood type? How would that violate the Hardy-Weinberg equilibrium? And that's what I'm going to let you guys think about on your own. And if you have any questions, just send me an email and think about that one and kind of talk through what the previous situations is at play in that scenario. So let's make the transition now to chapter 14, how all those little factors add up to give us new species. We see evidence in the fossil record of macroevolution. There's definitely plenty of evidence to support the production of new species. And in our own time, we're starting to see this new speciation within bacterial species. So this is something that there's plenty of evidence to support new species developing. Think of things like MRSA. MRSA is a developing species. And probably with antibiotic use, possibly even see more speciation with bacterial organisms. The reason we see that as far as time is concerned is what we were talking about in class. Bacteria divide and make a new generation every hour, where it takes us 20 years to make a new generation. So the time that's passed for bacterial generations in 20 years, we're talking a lot of generations. So we have all those other things, those microevolution events, the genetic drift or the mutations, things that help others thrive better than certain individuals in a given environment. What eventually may happen is that new adaptation, new gene, leads to enough changes that populations stop being able to breed with each other. And that really is the definition of a species, the ability to successfully breed with each other. Go back to the mule example that we talked about Tuesday. Mules definitely occur. We have horses and donkeys, two completely different species breed together, and they have offspring. And so you might be saying, well, bred with each other, but are they successful offspring? We talked about the fact that mules are sterile, so those aren't successful offspring. Same thing with ligers. Two different species, tigers and lions, breeding with each other. Very physically fit offspring are made, but they can't reproduce. And if you can't make more of yourself, then you're not a successful offspring. So species are able to successfully breed with one another and unable to successfully breed with other individuals of other species. And this leads to branching of the new species off of a parent species. Sometimes those branches are really far back in history, and we don't always know exactly where the branching point is because of that incomplete fossil record we talked about. But it's back there. It's something to appreciate with a common ancestor at a branch is that the branches off of that come from that parent, not from each other. A huge concern or argument with evolution is often, I didn't come a monkey. That's exactly right. We didn't come from the other apes. We came from a common ancestor that we share with the apes. So kind of think about that as far as speciation is concerned. So if speciation is all about reproduction, some of the things that were actually asked in class is, well, what makes species and what are barriers to reproduction? Here they are. There are extrinsic and intrinsic reproductive isolating mechanisms. Now, extrinsic we'll talk about with the allopatric, sympatric, parapatric speciation here in 1 second. Those are physical external barriers like a mountain range or a river or an ocean, something huge that keeps individuals apart from each other. A good example of this would be lions and tigers. Obviously, we get ligers. If they are together, they can mate. Obviously, they can have successful
fertilization. We do fertilization, we do get offspring. They aren't reproductively successful, but they are able to mate. Well, what's the big break there? Well, the big break is extrinsic there. Lions are in Africa and tigers are in Asia, and they're just not physically normally next to each other. So what are the intrinsic factors at play? There are 6 major intrinsic factors. There's ecological isolation, where maybe these individuals have an overlapping range. They do come in contact with each other every now and then, but their habitats are different. So even an ecological area might have distinct habitats within it, and we see that sometimes. There's also temporal isolation. These individuals just don't mate at the same time. May mate only at night or only during the day. Maybe some individuals mate during the spring, but some individuals mate during the winter. So there's something about their mating habits and timing that keeps them from being able to mate with one another. And there's also behavioral isolation. So all those things we talked about, like sexual selection, where you choose a mate based on a ritual. Think of birds. There's a lot of birds in one place at one time. Why don't they interbreed? Why are they separate species? Well, maybe there's a song that the female likes. Maybe the male needs to build a nest that the female has to check first. Maybe the male has to do a dance, or maybe the male has to have a certain color pattern. So there's that behavioral isolation with the choice of mates. There's also mechanical isolation. Sometimes the reproductive organs are different that they just don't fit, and your book has the Great Dane and like any tiny little Pomeranian. There's a mechanical isolation there. Those two are going to have a lot of trouble actually getting together to do the copulatory act to be able to get insemination and predation. Those are mechanical isolations.